Bismillah ar-Rahman ar-Rahim. Welcome back, friends. So we talked about the introduction of flagella, and we also talked about the structure of the flagella and types of bacteria according to flagella, and we also talked about the synthesis of the flagella. Now this is another video lecture which is also regarded with the cell biology, and in this video lecture we are going to talk about the function of flagella and the mechanism of flagella movement. So let me write function or mechanisms of flagella movement. Now what does it mean? It means that how flagella can perform their function, and when the flagella are performing their function, then on what direction the cell are moving. Okay, you know, whenever this flagella is rotate, okay, actually the flagella are in a rotate, they show their function, or they are moving in a rotation form. When the flagella are in a rotation form, when they are in a circular form, when they are moving in a circular form, so then the cell will move from one place to another place. Now this flagella, as you are watching, it is acting like a propel. Okay, so it is acting like a propellers. So it act as a propellers. What does it mean? It means just like in board, the propeller are used to move the board from one place to another place. In this case, the flagella are also act as a propel, just like in board, and it when it acts like a propel, so due to this way, the cell will move from one place to another place. Now the speed of this, whenever they are moving in a circular form and whenever they are moving in a rotation form, so the speed of such rotation will be 270 resolution per second. So 270 resolution per second will be their rotation, or we can say will be their speed of rotation. Now another thing that I must tell you about that, in case of monotrichous bacteria, all of you better know that monotrichous bacteria have only one flagellum. So in this case, such bacteria will be moved in a forward direction. What actually happened? Let me draw a structure of monotrichous here. So let's suppose if this is the monotrichous bacteria. Okay, now it have only one flagella. So please pay attention here. What actually happened? Now, whenever monotrichous bacteria is moving from one place to another place, this flagella will pull, means this flagella will be pulled in this form. When the flagella is pulling in this direction, so then the cell will be pushed to forward. Okay, so monotrichous bacteria will also means this monotrichous bacteria flagella will also show a clockwise rotation, means this bacteria will be moving in this form. So the flagella will be moving in this clockwise direction. Okay, so whenever they are moving in clockwise direction, so this bacteria will be moved in a forward direction. Whenever this direction is in opposite form, for example, whenever this whenever the flagella rotation are not in clockwise, when they are anti-clockwise, or when we can say that if the flagella are not if the flagella motion is opposite to the clockwise, we also call that anti-clockwise. So then the bacteria will show a tumble movement. Okay, sometimes they will move in upward, and sometimes they will move in a downward direction. Suddenly they will fall downward. So let me once again tell you what actually happened. Second case, we have that when the flagella show anti-clockwise rotation, due to this way, this bacteria, means monotrichous bacteria, will be moved, or we can say it show a tumble movement. Due to this movement, the bacteria will sometimes moves upward, and sometimes they suddenly move downward. Okay, now so this is about the monotrichous bacteria, that how it can move. Now in next case we have the Peritrackers bacteria. In this case, let's suppose this is again we have a bacteria cell. So in this case, as all of you better know, that Peritrackers bacteria have more than 2 or 3 flagella. So let's suppose one flagella, they will arises from that side, one flagella will arise from that end, one flagella arises from that area. So they have a lot of the flagella. Now, what actually happen during their movement? Whenever each flagella perform a counterclockwise rotation, for example, some of them are moving in this direction, some of them are moving in this one direction, some of them are moving in this one direction, and some of them are moving in this one direction, and some of means the flagella, all the flagella when show a counterclockwise rotation. Due to this way, the bacteria will be moved in a forward direction. Okay, so they will be moved in a forward direction. Why? Because they can pull the bundle of the flagella, means such bacteria can pull the bundle of the flagella to push the bacteria in a forward motion. Now, whenever all these flagella, as you are watching, whenever all these flagella have only one rotation, means they have only rotate in a clockwise, means just like in this one they will rotate. So then one thing you should have to keep in your mind, then such bacteria will show a tumble movement. Okay, so suddenly they will fall down. So that's how the Monotricus and Peritrackers bacteria can they can move. One thing that I must tell you about that in case of Peritrackers, not all these flagella will perform the function, but sometime what actually happen, these some of the flagella will deactivating and some of the flagella will be activating, but it is not occur usually. Okay, now in this case what actually happen, some of the flagella sometime not perform their function. For example, these, let's suppose these flagella, means these one flagella sometimes not perform their function. Instead of these flagella, this one area flagella will perform their function. So whenever they perform their function, so then the cell will move in the forward direction. But what actually happens, sometimes all the flagella will perform their function. So whenever all the flagella perform in their function, means when all the flagella are in a means counterclockwise. So due to this counterclockwise rotation, the bacteria will be means push forward, because they pulling all the bundle of the flagella behind. So due to this way, the bacteria will move forward direction. Now, but what actually happen in some cases, this flagella will be perform clockwise rotation. Due to this clockwise rotation, this bacteria will show a tumble movement, means they will suddenly fall down. Okay, on the other hand, if we talked about the means liphotrichous bacteria, which have the flagella at one end. Now, in liphotrichous bacteria, what actually happens? The flagella, as all of you better know, that liphotrichous bacteria are those bacteria which have the flagella on one end. So they have a lot of flagella on one end. Now, what actually happens during their movement? All the flagella will be in a clockwise. Okay, so they will be in a clockwise rotation. They will be in a clockwise. So whenever these flagella are rotate in a clockwise, so the cell will be move forward. Okay. So, just like Monotricus, the same function will be the means liphotrichous bacteria as well. But this liphotrichous this Peritrackers bacteria movement is not clearly understood. Why? Because sometimes they show tumble movement, sometimes they show forward movement, sometimes they show backward movement, because they have a lot of flagella. So the movement of this this bacteria is not clearly understood. Now, so that's how the bacteria can move. Now the second thing which the question which arise in our mind, that how actually the movement is possible, means how these flagella can rotate. This is our questions. So the rotation of this flagella will be possible by this rotor portion. Means rotor portion, all of you better know that they contain fly proteins, fly M, fly N, fly G proteins, SM ring. All these rings, I already explained the structure. So if such bacteria contain these rotor portion, which have rotor portion means SM and fly proteins. So due to this portion, this rotor portion will be rotated. Whenever this rotor portion are rotated in clockwise or anti-clockwise or counterclockwise, due to this rotation, the flagella will rotate. Okay, the flagella will be performing their function. Without this rotation, the flagella will not perform the function. The first thing. The second thing is that whenever this rotor portion is rotating, so similarly the rod will also rotate. So whenever the rod is rotated, so then the flagella will rotate. But keep one thing in your mind, not only a PL, but also this we can say that stator portion is in a fixed and in a stationary form. But here another question which are also arises that what actually happened, that how this rotor are rotated. If I already to tell you that what actually happened, this rotor portion are rotating, that's why the flagella are rotate. But how this rotor are rotate? What molecules are actually present inside the cell or outside the cell that are responsible to rotate this rotor portion? Now let me explain. What actually happened, we have a lot of proton, at that area, as you are watching, at that area, means at stator point. So this, all of you better, this is the mode A and this is the mode B area. Okay, so this is the mode B. Now in this case, what actually happened, at outside of the cell, as all of you better know, outside is the higher portion and inside of the cell it is the lower portion. So outside the cell there are a lot of sodium. At that area, means at the stator area, with the stator air, there are a channel, we call that proton channel. So let me write here at that area. Let me draw a separate here. So let's suppose this is the stator area. Okay, let me draw here. Let's suppose this is the stator area and this is the, let me change the color, and let's suppose these are the mode proteins. This is, let's suppose, mode A and this is, let's suppose, mode B. Okay, so this is A and this is B. Now at this specific area, as you are watching, there are aspartic acid are present. So let me write aspartic acid. Now there are aspartic acid are present. What actually happened during means the so here as you are watching, at that portion, at that site there is a channel, as you are watching. At that site there is a channel, we call that a proton channel, or some holes are present. At this specific area there are the sodium molecule which are present outside of the cell, which is the higher concentration. The sodium molecule, let me draw with a black marker. These are, let's suppose, the sodium molecule. So this sodium molecule will passes from this aspartic acid. So they will be bind to this aspartic acid. So when two molecule of the sodium or proton will bind to this aspartic acid, so what actually happen? First power stroke will be happen. So during first power stroke, the mode A will be downward and the mode B will push upward. So during this way, this means this rotor portion will be rotate. They will be rotate in a first power stroke. So they will be a rotate. So what they will be a rotate. Now whenever these sodium are released into the cytoplasm area, so then a second power stroke occur. In second power stroke, what actually happen? This one means this mode A will be move upward and this mode B will be moved downward. During this way, a second power stroke occur. Okay, again the cell will means again this rotor portion will rotate. Okay, so let me once again tell you that how actually the motor portion are rotated. During this way, this mode protein contain aspartic acid. This aspartic acid are responsible to attract the sodium molecule or proton molecule. Once they attract the sodium molecule, so the sodium molecule once connected to this aspartic acid, so then they will cause a first power stroke. During first power stroke, the mode A protein will push downward while the mode B protein is pushing upward. During this way, this rotor portion will rotate. They will rotate first. After that, these sodium molecule will leave this aspartic acid. Once they leave the aspartic acid, what actually happen? After that, another second power stroke occur. In second power stroke, this mode A will be pushing upward and mode B will be pushing downward. During this way, again this rotor will rotate. Okay, so that's how the rotor are rotated, and the process is continuing until this we can say sodium are coming. Okay. Now there is another question arise in our mind, that bacteria are okay, the bacteria is moving, but the bacteria is moving in a swimming form. Okay, whenever the bacteria are moving, so they are actually moving in a swimming form. What actually happened in this case, these bacteria are present in sometime in air. These such bacteria are present in the water. Such bacteria are present on the, we can say, on the surface area. So whenever the bacteria are present on that area, so then the bacteria will be swim on such area. Okay, the bacteria is not actually showing some other movement. They will be showing a swimming like movement. Okay, and if we see the speed of such bacteria, means whenever the bacteria are moving from one place to another place, so the speed of such bacteria will be 2290 micrometer per second. Now what does it mean? It means that they can cross 100 cells. So they can cross 100 cell per second. Whenever this flagella can perform their function, so then the cell will move from one place to another place, and during this way they can cross the 100 cells. Okay, and this speed will be means 2290 micrometer per second. So now we have a bacteria, we called it spirochetes. Spirochete bacteria are usually, they can usually move, or they can swim in viscous or water or fluid like area. Whenever they can rotate their flagella, or whenever they can rotate their axial filament or spinning this filament. Now such movement we call that gliding movement. Okay, let me once again tell you, spirochetes is a kind of bacteria. Such bacteria are usually present in a mud, viscous, or we can say jelly like substance, or we can also say that it is usually present in a fluid material. Now due to this, during this time, what actually happen, this bacteria will show a gliding movement. What does it mean? It means that they will be swimming in such area, sometime in a fast motion and sometime in a slow motion. But during this time, the flagella will be spinning. When the flagella is spinning, so then they will perform their motion, or when then they will perform the swimming motion. Now such movement, as we call that, means gliding movement. Gliding movement. Okay, such gliding movement are usually present in cytophage, and we have myxobacteria, and we have also cyanobacteria. Okay, so that's how the flagella can perform their function, and that's how the mechanism action of this flagellar movement. I hope you understand about this video lecture, but if still you have any kind of questions in your mind, so write it into a comment. Inshallah, I will give you answer as soon as possible. So if you like this video, so make sure to hit the like button, share this video, and also subscribe the channel, and stay tuned, because in the next video lecture we are going to talk about the difference between gram-negative flagella and gram-positive flagella. Okay, and then we are going to talk about eukaryotic flagella as well, because eukaryotic flagella function and motion and their structure are also different from this one flagella as well. Okay, so please stay tuned and continue to watch the video. Thank you so much.